My name is Agnete Kirkeby and I'm a fellow at the Wallenberg Center for Molecular Medicine at Lund University. In my group, we use stem cells to produce human neurons in the lab. We do this because many people around the world suffer from neurodegenerative disorders such as Alzheimer's disease, Parkinson's disease, epilepsy, dementia. And when the adult brain loses neurons, such as in these cases, it cannot repair itself. We use stem cells in the lab to produce new neurons, which can be used as a regenerative therapy to replace the neurons that have been lost in the brains of these patients. We can use the stem cells both as a model to study how the human brain develops, and we also use the stem cells to produce new neurons for transplantation. Using stem cells in the lab, we can produce any neuron desired. We produce high-purity cultures of human dopaminergic neurons, which are normally lost in the brains of Parkinson's disease patients. We've shown through work in animal models that the neurons are functional and can repair the brain and restore motoric function in the animals. And we're expecting this to go into clinical trial during next year. The goal of our research is to understand how the human brain is formed and how it functions, and how we can repair it by generating new cells in the lab. Our aim is to produce regenerative therapies for various different types of neurodegenerative disorders to alleviate patients from the use of daily medication.